Okay, let's go ahead and get started. Thanks for joining me. Let me go ahead and share my screen. I want to ask a question first. Was anyone with me last week when I did this? Okay. So I have some FRQs from the past 3 years, 2017, 2018, and 2019. Have you guys all worked on them? Can you tell me if you've worked on 2018 or 2017 yet? Can you guys kind of, anyone, let me know in the chat or, um, you know, here. Otherwise, we'll go through a little bit from all three of them. All right. Well, this test is going to be way different than any other test you guys have ever had. You're going to only have 45 minutes, and it's only going to be 2 FRQs. The first one, 65% of your score. And I got an example what it's going to look like. Same thing as the first one we're about to take a look at. But what's going to happen here is you have a scenario, definitely going to be given a data table and complete a few parts. Describe and explain the concepts or processes that's going on. Identify and justify an experimental design procedure. Analyze some data and make and justify predictions. Now, question 2 is very similar, except you don't have a data table to analyze from there, but you're still going to describe some sort of concepts or processes, explain what's going on, predict what happens if you mess with it. So that's kind of one of the new things, and then basically support why you said that. Now, these 6 units are still going to be on the exam. The chemistry of life, dealing with macromolecules, the why DNA holds itself together, and polarity, stuff like that. Cell structure and function. You guys know the different types of the differences between prokaryotes and eukaryotes, and even differences between plants and animals. And you're going to need to know the functions of all the organelles. Cellular energetics includes photosynthesis, cellular respiration, stuff like that, transport, the communication and cell cycle, basically how cells signal to each other, what's going on in the cell cycle, mitosis or meiosis. Heredity is actually more meiosis here, and genetics. And then gene expression and regulation, and basically how the transcription translation process occurs, and anything that might stop that. So no natural selection, no ecology. I know those might be some of the easier ones, but not going to see that, so we don't have to study anything there. So let's get started with the first type of FRQ. So I'd like you guys to take out a pen and paper, because I want you guys to take a couple minutes, try to write down some of these answers, and then we could talk about your responses, because we got to get really good at not just understanding what the answer is, how to write it, what they're asking for. And so let's get practice with that. So we'll go ahead and read this first, and then once we get to A, take a minute to write some ideas down. So first, in flowering plants, pollination is a process that leads to the fertilization of an egg and the production of seeds. Some flowers attract pollinators, such as bees, using visual and chemical cues. When a bee visits a flower, in addition to transferring pollen, the bee can take nectar from the flower or use it to make honey for the colony. Nectar contains sugar, but certain plants also produce caffeine in the nectar. Caffeine is a bitter tasting compound that can be toxic to insects at high concentrations. To investigate the role of caffeine in nectar, a group of researchers studied the effect of 0.1 millimolar caffeine on bee behavior. The results of an experiment to test the effect of caffeine on bees' memory of a nectar source are shown in Table 1. Okay. Now, on axes provided, construct an appropriately labeled graph to illustrate the effect of caffeine on the probability of bees revisiting a nectar source. Okay, we have memory, average probability, plus or minus 2 standard errors. And we have a two-way table, control caffeine, 10 minutes and 24. So go ahead, everyone take 1 minute draw what you think might be the appropriate graph and label it. And then if anyone wants to either explain what they did in a minute or type it into chat, we'll go over that. So take 1 minute and do that, please. Anyone want to try A? Were you guys able to hear me the whole time well? Oh, can you guys hear me? Okay. All right. Anyone let me know what they think, what type of graph might be best to use for A? Definitely a bar graph. And we probably have to make a couple of them. So we have control. We could say this is somewhere 72. And then that's 10 minutes. And then we could say this is 24 hours. Caffeine, 83. And then 78. Something that looks like that would be fine. Now, anytime we do a graph, we definitely have to explain what's going on. So we could write how we have control and caffeine on the bottom. We're supposed to label everything. So let's just pretend we did it quickly, 0.7, 0.8. And we have to explain the differences between here's the 10 minutes, 24 hours, 10 min, or we could have done it somehow in a legend, 24 hours. So we need all that, but that's still not going to get us all of our points that we need here. Does anyone know the last thing they're looking for to describe the stuff graphically? Axis labeled is okay because that's kind of controlling caffeine. Title, they're not really going to take away the title, even though I definitely recommend you put one, but they're definitely going to take away points for the error bars. So always, if they give you some sort of plus or minus, explain or show that there. So if this is 0.72 plus or minus 0.9, go ahead and go up and down a little bit. Plus or minus 0.07, so that's not that big. And then these guys, same pretty small error bars. So the error bars were one thing that they're definitely going to look for if they ask you to do that, or if they show these, or have a confidence interval. So a few different ways that that's going to pop in. So nice one there. So, okay. So most people, I'd say, would have got 2 out of 3 points. The error bars, many people would have forgot those, but make sure you don't in the future. So let's go for B. Based on the results, describe the effect of caffeine on each of the following: short-term memory of a nectar source and long-term. So if you guys would like to take a second, type it into chat or let me know, we could grade your answers. Any ideas for short term? Okay. Caffeine does not affect short-term memory loss at 10 minutes. What makes you say that claim? What is the kicker? Okay, so got a couple good answers. One of them looks really good, Benny, but I don't think they'll give you the points for that one. Now, what they're looking for is you cannot say anything is different in the field of statistics, and there's kind of the first question is going to be partly statistical if the bars overlap. So if we take a look at the short-term 10 minutes, 0.72 plus or minus 0.09 gives us an interval 0.63 to 0.81. So 0.63, 0.81 for the first guy. The second guy, 0.76, 0.9. Now, although the intervals are different, there's overlap. If there's any overlap at all, the 0.76 through 0.81 would be the overlap, then you cannot say that you found a statistical difference. It looks barely, but we can't even say barely. We have no evidence to suggest that. So that one's there, and it has to be, yes, explain the margin of errors and how there is overlap, and we definitely wanted to do that better. If I drew it a little more carefully, we could have seen it, but if you also pull that out and show the interval that we expected in, we could say we don't predict a difference there. We don't have enough evidence. But what about long term, 24 hours? Yeah, quite a hugely significant difference. Those bars don't overlap whatsoever. In fact, they're very far apart. So perfect. A and B are the first two questions there. Next, this one's going to be pretty important. So for sure, everyone, I'd like to see if you can type it into the chat so I can kind of grade as many as possible, even though they might be similar. So go ahead for C. Design an experiment using artificial flowers to investigate potential negative effects of increasing caffeine concentrations in nectar on the number of floral visits by bees, identify the null and appropriate control, and predicted results that could be used to reject the null. Okay, take 1 or 2 minutes. Very important question. So anyone got the null so far, at least, if they want to type that in? Okay, there we go. So we have 2 flowers. Exactly. So both of those ways of expressing the null hypothesis are correct. I feel maybe the second one is a little bit cleaner, but you're still getting points for both of them. So that's fine. As long as you say there's no change, there's no effect, and then explain it in the context of the problem. You can't just say there will be no effect. That's not going to slide there. But you guys did that one very well. The control treatment, let's see if you guys had that in. Okay. So that second one, where you say we have different control groups, that's usually not ever going to happen. We usually only want one control group. Sometimes there's not any control groups if you're comparing 2 different populations to each other. But if you can have a control where you do not do anything to one group and you do something to the second, we'll have that. So there's one control group, which is the first one you said, the one which is going to have no caffeine present. The others are called experimental groups. So you'd have to clearly state that those are not the control groups. But yeah, so control flowers with no caffeine. The experimental groups would be flowers with caffeine. You definitely could have said these different levels of experimental groups. They're not going to take away points for making it a nicer experiment. But we definitely had to say those were experimental. And then predicted results. If we want to reject the null hypothesis, we'd have to say, yes, the number is different at increasing caffeine amounts. So that's perfect null. So I know this one it's definitely easy to get very close to the answer, but to hit the points, all of them. Anyone have any questions on how they have to phrase it or anything like that? Not math based that much at all, but with the first data one, they're definitely, and if you look back at all the FRQs, there's usually at least one sort of data statistical question in them. So a little bit there, but I definitely think, yeah, at most a little bit. I don't think you'll have a full one like this one, but you could definitely have a question there. Okay. And remember the first board says describe, so we definitely will be getting that data table. And yes, they like to use confidence intervals and statistics at least once per exam. But then let's go ahead for D, finish off this question. Researchers found that nectar with caffeine tends to have a lower sugar content than nectar without caffeine. Plants use less energy to produce the caffeine in nectar than they do to produce the sugar in nectar. Propose one benefit to the plants that produce nectar with caffeine and a lower sugar content, and one cost to bees that visit the flowers of plants that produce nectar with caffeine and the lower sugar content. So quickly, we'll just talk about this one, I'd say. Well, it's energetically favorable. They would need to consume less energy. Yeah, more pollen transfer, definitely another benefit. So good one there. A cost, well, they're getting less sugar every time the bees go in there. They have to go do more frequent visits, and it was toxic to them. So pretty straightforward of a question for D, worth a very few amount of points, by the way. Definitely saving energy. Yeah, those will definitely get you points there too. Okay, so let's go ahead to the next question. Let's get at least a few good ones in. 3. Gibberellin is the primary plant hormone that promotes stem elongation. GA3H is the enzyme that catalyzes the reaction that converts a precursor of gibberellin to the active form of gibberellin. A mutation in the GA3H gene results in a short plant phenotype. When a pure breeding tall plant is crossed with the pure breeding short plant, all offspring in the F1 generation are tall. When F1 plants are crossed with each other, 75% of the plants in the F2 generation are tall and 25% of the plants are short. The wild type allele encodes a GA3H enzyme with alanine, a nonpolar amino acid at position 229. The mutant allele encodes a GA3H enzyme with threonine, a polar amino acid at 229. Describe the effect of the mutation on the enzyme and provide reasoning to support how this mutation results in a short plant phenotype in homozygous recessive plants. Okay, couple minutes. We have describe the effect of the mutation and provide reasoning to how it results in short. Yes, the amino acid substitution changes the shape of the protein. And the mutation leads to decreased gibberellin production. Okay, good. So for the GA3H enzyme will lead to shorter plants, you have to explain why. And if you look above, there was a great answer. It's because that gibberellin explained that is a primary plant hormone that promotes stem elongation. So kind of rephrase their beginning. So if that promotes stem elongation, we want more of that. And if you don't have more of that, you're going to be short. So a little bit more clear there. So that was a good one, Annalise. Here, the mutation halts the height growth of the plants because the protein, well, let me type that in here, is unable to activate gibberellin. Yes, it's unable to activate it or use the active form. They're definitely going to give you points for this one here. So those are a lot of good ones. Now go ahead, try B. Using the codon chart provided, predict a change in the codon sequence that resulted in that substitution of alanine for threonine at 229. Okay, that one looks good. I definitely say an A was substituted into the first position, and then you could have got that answer fully correct. I think they would have gave that a partial. or explain this exact thing right here, GCA to ACA, or you don't have to say GCA because it could have been GC or yeah, GC anything to AC anything. So that one wouldn't be complete either. So yeah, just kind of just say A was substituted in the first base of the codon instead of G. That's the most clean answer. That first base, that position is pretty important. And then basically, if you'd like to represent that you don't care what that amino acid is, you could kind of, what that base is, you could just put N for nitrogenous base. Like if you put GCN to ACN, they would know what you're talking about. So, but definitely at least getting partial credit all over there for everyone there. But full credit is a little tricky. Make sure first base. Last question, describe how individuals with one heterozygous or two homozygous copies of the wild type can have the same phenotype. Okay, anyone have that? Exactly. So a couple good answers. We have there is enough active enzyme produced from one wild type dominant allele that would suffice. So here it says since the allele for short plants is recessive, you can have both of them that have the same phenotype. You kind of rephrased the question there. You didn't really describe how. So they would not give you points for that one. You'd have to definitely explain how one of them can cover for the other if it's a mutant and produce enough to be efficient and enough to make that change. So it still codes for some, maybe not as much, but you definitely don't want to say it would be halted, because even though it might be slightly shorter, it might not be. They definitely might produce enough gibberellin there. Heterozygous show the dominant. So again, that one doesn't hit the description on how they can have the same phenotype. So it has to be enough active enzyme is produced or enough gibberellin is produced in the presence of one wild type. So that had to kind of be that direct for that one. They didn't really want anything else there. Questions with anything on problem 3 before we go on? Okay, let's go on. Problem 4, this is going to be something similar to what we have. Remember, we're going to have to identify what's going on, justify, explain. So there would be something similar to this. So let's go ahead and read it. The common bedbug is a species of insect that is becoming increasingly resistant to insecticides. Bedbugs possess several genes suspected of contributing to the resistance, including P450, ABCA, and CPS. To investigate the role of these genes in insecticide resistance, researchers deleted one or more of these genes in different strains of bedbugs, as indicated in Figure 1, and treated the strains with the insecticide beta-cyclothurin. Each strain was genetically identical except for the deleted genes and was equally fit in the absence of that thing. The percent survival of each strain following the treatment is shown in Figure 1. Identify the control strain in the experiment. Use the means and confidence intervals in Figure 1 to justify the claim that ABC8 is effective at providing resistance to beta-cyclothurin. So go ahead, what is the control in this experiment? And justify the claim that ABC8 is effective. So the control is the first strain. Perfect. That's all they needed there. And that's because they are wild type for each of them. And then ABC8 is effective because once it was removed, the strain had the lowest percent survival rate. So that's okay, but it's not going to get you the full points because it said use the means and confidence intervals to justify that claim. So again, this one did not use the means. So, well, you did use the means, so I'll give you partial credit. But what about the confidence intervals? I want to give that a try. So again, the error bars. So we didn't read this together. Percent survival of 5 strains. Plus indicates gene is present, minus is deleted, and error bars are the so again here, no, they're kind of again asking about overlap. Since there's no overlap between the first strain and the third strain's error bars, we can conclude without a doubt, or with at least 95% confidence, that once you remove ABC8, you are diminishing their chances of survival. Like, even though, let's take a look at 1 and 4. We can't say 1 and 4 are different. Yeah, we have different means, but we didn't take enough data to have small enough error to say that, you know, those populations truly are different. So since there's overlap between 1 and 4, we can't claim that CPS does anything. But again, you wanted to know that you wanted to state overlap of the bars. And that allows you to state for sure there is a difference. Okay, let's go here. P450 encodes an enzyme that detoxifies insecticides. ABC8 encodes a transporter protein that pumps insecticides out of cells. CPS encodes an external structural protein located in the exoskeleton that greatly reduces the absorption of insecticides. Based on the information and data in Figure 1, explain how a deletion of both P450 and ABC80 results in lower survival in bedbugs than a deletion of CPS only. So CPS did have a lower, but again, we didn't know it was much lower because of the error bars. But when you remove P450 and ABC8, we see it for sure. Now, based on what they do, explain a possible reason why. So go ahead and take a couple minutes there. This one's another pretty important one, worth a few points, this one explanation. Any attempts? So the removal of both P450 and ABC8 makes it harmless to the bugs. Actually, it lowers their percent survival, so it's extremely harmful. So that one, nice attempt. I don't think they'd give credit for that one. So let's take a look at it. P450 detoxifies, ABC8 transports insecticides out of cells, and CPS greatly reduces the absorption. Okay, let's take a look at that answer. So P450 and ABC8 work together to detoxify and pump insecticides out of the cell. A deletion of both of them would not lower the survival. Well, deletion of both of them would lower the survival because the insects, because it's not. Yeah, okay, perfect. There we go. A deletion of CPS would not encode. So, perfect. So that's a good one there. Now we kind of definitely want to explain how even if they absorb some, if those detoxifiers can work and pump it out of the cell. So what if you absorbed it? But even if, let's say you only absorb a very, very little amount. If you only absorb a very little amount, but you're never, ever, ever able to take it out of your cells, over time it will kind of grow pretty quickly and eventually, cause a disaster there. So if you still absorbing it but never getting rid of it, it'll eventually kill you. But if you're absorbing it but getting rid of it fast, maybe you'll be able to get rid of it fast enough. And here it definitely shows, yes, they're able to get rid of it at a fast enough rate that their survival from removal of just CPS is much greater than the removal of both of the other ones. That one was a tough question. Any questions there before we go forward? Okay. Take a look at this one. So cell with DNA damage, an individual nucleus after an electrophoresis. Intact DNA remains in the head and damaged DNA is in the tail. So we have a common assay to detect double-stranded breaks in DNA. And let's read this. A common assay is a technique used to determine the amount of double-strand breaks in DNA or DNA damage in cells. The nucleus of an individual cell is placed on a microscope slide coated with an agarose gel. An electric current is applied to the gel that causes DNA to move, electrophoresis, and the DNA is stained with the fluorescent dye. When viewed using a microscope, undamaged DNA from the nucleus appears as a round shape, the head, and the fragments of DNA extend out from the head, the tail. The length of the tail corresponds to the amount of damage in the DNA. To explain the movement of DNA fragments in the common assay, identify one property of DNA and provide reasoning to support how the property contributes to the movement during the common assay technique. Go ahead, give that one a try. Okay, perfect. That's one of the two acceptable answers. And okay, you guys definitely hit one of the two, and they'll give you full points for that one. Excellent. The only other thing that would have got points is if you said smaller fragments are able to travel further, and that's why you see that movement. So you could have either said smaller fragments would be able to travel farther, or you can state just in general how DNA, why it's even moving that way in the first place, left to right. But then these are moving left to right a little bit slower than the smaller guys are in the tail. So either or, full points across the board there. In a different experiment, cells are treated with a chemical mutagen that causes only nucleotide substitutions in DNA. Predict the likely result of a common assay for this treatment. So that chemical mutagen that we were talking about damaged DNA. But here we have a chemical mutagen that says it damages it by causing only substitutions. What's the likely result? Well, if they are pretty much all the same, exactly, you'd have a round shape that does not have a tail because you wouldn't have any damage. They'd all be the same length, even if they had one switch someplace. So it'd be intact in the same length, and you'd only have one circle. Perfect. Okay, let's see what other good ones there are. Perfect. That one would work. The answer would work well too. So let's go ahead and give this one a try. Cystic fibrosis is a genetic condition that is associated with defects in the CFTR protein. The CFTR protein is a gated ion channel that requires ATP binding in order to allow chloride ions to diffuse across the membrane. In the provided model of the cell, draw arrows to describe the pathway for the production of a normal CFTR protein from gene expression to final cellular location. Okay, so gene expression basically begins where? Well, gene expression begins in the nucleus first. You have to get your mRNA, well, your gene was DNA, and then you got to copy it into mRNA, and then we have to deal with that. So we have nucleus, definitely nucleus, ER, Golgi, somewhere to the plasma membrane. Absolutely. The Golgi actually would create the vesicle, which is perfect. Yes. So we have this chain of events right here. Now, not every protein that you make needs to go into the ER. A lot of proteins, once they leave the nucleus, if they're very, very small or if they have another location, can be made by the ribosomes freely floating in the cytoplasm, like all proteins in the prokaryotes, since they're transcribed and translated both in the cytoplasm with the freely floating ribosomes. But this one is a membrane protein. Membrane proteins must be modified by the ER, must be modified by the Golgi. So there's no way around that for any sort of membrane protein. In order for it to stick in the membrane, an endoplasmic reticulum must add the lipids that are required for it to fit in nicely and kind of just, stick with the nonpolar aspect of the plasma membrane and embed itself right inside of it. So we have that arrow, or they kind of would have gave you points if you drew the straight arrow that crosses through everything, at least if it goes here. But I think you guys handled that one very well. Identify the most likely cellular location of the ribosomes. Definitely, we talked about that already, gave the answer away. For sure, the rough ER. No, that one you could just draw arrows, because read the question a second or third time if you're not sure, and they're never gonna grade on more than they ask, but sometimes they have a word in there that you didn't read well and you forget to answer fully, but it just said draw arrows. That's full points, those arrows. And then yes, the rough ER. And then if they asked why, we could say because it's a membrane protein and it needed the lipid. So I have a feeling, since there's only 2 questions this time and they have to blend more units together in single questions, they'd ask you why, and then you have to explain something like that. Now this one's a little tricky. So think about this one. Identify the most likely cellular location of that protein. Okay, why do you guys think it's going to make it to the plasma membrane? If it has amino acid substitution in the ATP binding site. So just in case why was a question next time. What's your justification? It is one, but I wouldn't say that's a good enough justification for how it made it there or why it didn't get stopped before it got there. Okay, so this one, you definitely have to say how there was no mutation that caused a change in the ability to make the enzyme. So it's still going to get exported from the nucleus. It's still going to get packaged by the ER and the Golgi. They don't know that it is defective because that ATP binding site is only going to affect its functionality and never affect its ability to have been made in the first place. If they said it has a substitution in the binding site which crippled it, or if it had a substitution for methionine or a completely different thing, then maybe. But if it was just a small substitution, it would still get made and sent out there. But if that substitution is in the binding site, it would become ineffective there. So that's a good one. Let's go to the next question. Okay, didn't do too much genetics today. In the tongue sole fish, sex is determined by a combination of genetics and environmental temperature. Genetically male fish have 2 Z chromosomes. Genetically female fish have 1 Z and 1 W. When fish are raised at 22 degrees Celsius, ZZ fish develop into phenotypic males and ZW into phenotypic females. However, when fish are raised at 28, the Z chromosome is modified, denoted as Z star. Z star W develop as phenotypic males that are fertile and can pass on the Z star chromosomes to their offspring, even when offspring are raised at 22. A cross between a ZW female and a Z star Z male is shown in the Punnett score below. Predict the percent of phenotypic males among the F1 offspring of the cross shown in the Punnett square if the offspring are raised at 22 degrees Celsius. Perfect. 75%. We have male, male, phenotypically male, female. At least one Z or Z star chromosome is necessary for the survival of the fish. A researcher crossed two fish and observed the 2-to-1 ratio of males to females among the offspring. Based on the information, identify the genotype of the male parent in the cross and describe one fitness cost to the female of mating with this. Don't worry about fitness cost. Nothing about evolution is going to be there. But what would that genotype of the male parent be? If they had a 2-to-1 female to males. So we know we have a female. So female had to have been ZW. And if it was a male parent, two options you can have, or three options. You can have ZZ, bunch of stars, just one star, or you can even have one star and a W and still be that. So you have Z star W, and if we had that Z star W, that would create phenotypically males right here, males right here, here's females, and here's dead fish. So two to males to one female to one abomination that dies because it doesn't have enough genes to basically develop. So 2, 1, 1, perfect. Z star W was the right answer. We could have tried all of them, and that would have been the only one that had it, but you had a kind of an idea of beforehand that that's how they would die. So good thing you checked that one first. Okay, a few more questions. Let's see. Okay, let's actually do this one. I think this is the best one. So acetylcholine receptor proteins are found at the synapse between neurons and skeletal muscle cells. Acetylcholine released from neurons binds to a specific site on the receptor proteins, which causes an ion channel in the receptors to open and allows sodium ions to enter muscle cells. The resulting depolarization of muscle cells initiates muscle contractions. Another molecule, nicotine, can also bind to certain types of AChR proteins and activate the receptors. A researcher is investigating two different types of AChR proteins, 1 and 2. To determine which stimuli activate the receptors, researcher exposes muscle cells expressing the different types of receptor proteins to stimuli and observes the results indicated in Table 1. Plus for acetylcholine, plus minus for nicotine. Describe the difference in structure and function between AChR type 1 and type 2. Okay, take a minute there. There'll be 1 point for each. So they differ in shape, so there goes your point for structure. And they bind to different things. But what is, and then function, you kind of have partial credit for the function, but they're not going to give you full points. How do their functions differ? Or what is the relation of their functions? Okay. Again, that's structure. You'll get the full credit for that. For function, they kind of want you to state that there is really, in terms of function, no functional difference besides the ability to differentiate acetylcholine and nicotine due to their shapes. They perform the same thing when activated. So when activated, not kind of depending on what activates them, but when activated, they perform the same function. So that's kind of something that they wanted to make you state. Okay. Again, yeah, this one I think was a harsh way to grade it. I probably wouldn't have gone this one because I wouldn't have explained that the function is the same response when activated, but still activated by different things. So make sure function, what does it do when it does get activated? Definitely going to be points. So tricky one, but yeah, think of that when you're thinking of function. Because although the structure dictates the function, we want to clearly state function is the same. Okay. Acetylcholinesterase is an enzyme that breaks down acetylcholine in the synapse. Describe the effect of inhibiting acetylcholinesterase on the muscle cells with AChR type 2. Give that a try. Okay, this accumulation of acetylcholine will result, leads to more muscle contractions. Perfect. So since it does not have the ability to break it down, or since you inhibit acetylcholinesterase and you're not breaking down acetylcholine, you build up acetylcholine and lose control of the muscle cells, or yes, you guys both getting full credit for that answer. So you definitely have to explain how there's a buildup and how and what that buildup does. Excellent. All right. So that's about all the time we have for today. I wanted to ask if you guys had any last questions about any of the problems we went over today or the general structure of the exam before we get finished. So any last questions? if there aren't any last ones. I definitely wanted to thank you guys for joining me. I'm very happy to help you guys. I'd like you guys to do as best as you can on that test coming up. Hope you don't feel too stressed out. Make sure you bring well, make sure obviously you have your textbooks, but what I want you to do is if you're going to answer a question, go to the picture that would be described there. So if you're dealing with meiosis, go straight to the meiosis pictures. Or if you're doing something with the cell structure, go straight to the pictures of the cell. Now have just enough information to jog your memory. So don't try to do everything with what you have in your head. It's an open notes. Just quickly open to the page. Pictures are going to be extremely helpful. I definitely wanted to offer a discounted rate. My normal rates are $100 an hour, but for everyone who joined today, I'd like to offer a lower rate of $70 an hour. So if you do decide to work with me preparing for the test, if you do send me a message, I'll give you lower rate. So just know that you're not going to be paying what you see there. Other than that, thanks again for joining me, and good luck and have a good rest of your day.